All right, so I'm going to go into a little bit more detail about the first part of photosynthesis now. You should know that that's the light-dependent reaction by now, and what we're really going to do is dive into a lot of the nitty-gritty of what's going on in the light-dependent reaction. So what I've done is looked at the list of understandings on page 389 and wrote out the ones that you need to know for the light-dependent reaction. And that long list on page 389 includes light-dependent and light-independent reactions. But again, this is on the light-dependent reaction. So I'm going to go through this and make sure that you understand and can read what I've written here. These are the understandings that you should be able to walk away with by the end of the video. And if not, you should supplement by reading in your textbook or rewatching or going to YouTube, because you probably should be watching this in Edpuzzle, but going to YouTube and rewatching this video. So the first is light-dependent reactions take place in the inner membrane space of the thylakoid. So it's telling you the location of the light-dependent reaction. You're going to have to know the drawing of it, and that's really going to help with your understanding for the other things. Reduced NADP and ATP are produced in the light-dependent reactions. And so when we talk about reduced, what you should be thinking about is, let me write this as redox reactions. And that I know is something you did in chemistry. So redox reactions are going to be really important to make sure you understand. And there's a little shortcut, I don't know if you learned it in chem or not, oil rig. And we've got oxidation and reduction reactions. And this is all really about electrons and where electrons move. So something that is oxidized is losing an electron, and something that is reduced has gained electrons. So oxidation is loss and reduction is gain of electrons. So when we talk about reduced NADP, we're talking about NADP that has gained electrons, and ATP that have gained electrons through the light-dependent reaction. We also need to make sure we understand that the absorption of light by photosystems, which maybe is a new word for you, generates excited electrons. Photolysis, which may be a new word, I don't think it is, because I think I mentioned in a previous video, the photolysis of water generates electrons for the use of the light-dependent reaction. So the water is the source of the electrons for the light-dependent reaction. And if the transfer of these excited electrons occur between the carriers, between these things called carriers, in the thylakoid membrane. Those excited electrons from photosystem 2, which is an important word, photosystem 2, are used to generate a proton gradient, yet another term. An ATP synthase, a very specific molecule, a specific enzyme or protein, in the thylakoids generate ATP using the proton gradient. And then excited electrons from photosystem 1 are used to make NADP. So there's photosystem 2 up here, and then photosystem 1 that comes later. And that's kind of a lot of words for this process. So let's talk a little bit about some context here. So what I've drawn is a chloroplast. I've not labeled it as a chloroplast. So this is not a plant cell, it's a chloroplast. The chloroplast has an outer membrane and an inner membrane, and then there are these thylakoids, which you've drawn in a previous diagram. This looks a little different. I've zoomed in quite a bit, so we've got these squished pancakes. This one's kind of folded. And we've got water in the equation here. And so what water can split into is 3 different components. We've got the hydrogens, we've got some oxygens, and if you remember that the structure of water is kind of like, if you remember the Mickey Mouse shape, there are these covalent bonds that hold these 2 hydrogens to the oxygen. And those covalent bonds are shared electrons. Those electrons can be released when the molecule is broken. So through photolysis, when this water molecule is broken, electrons are released, which are going to be really important because we've already talked about electrons getting excited or having more energy. And we've also talked about oxygen gas being released. And I've also mentioned the H in the hydrogen or the protons, right? A hydrogen cation has lost its electrons. The electrons are over here. And all that's left in a hydrogen atom, if it loses its electrons, is a single proton. There are no neutrons involved. So these are just protons, right? So this is going to come into play with the proton gradients. But here in the thylakoid, we have our light-dependent reactions, and light's coming in, water's involved, and oxygen comes out, hydrogen leaves, and so do the electrons. I do want to point out that the chloroplast is a double phospholipid bilayer. So each one of these lines represents a bilayer. The cell, kind of the same thing as a cell membrane, but there are two of them here. And a good question that you should consider is why there might be two phospholipid bilayers. But that being said, these electrons are going to then, if you go back to your list of objectives, they will reduce NADPH. So NADPH has electrons added to it, NADH has electrons added to it, and it turns into this other molecule called NADPH. Another electron goes to this up here, and I apologize for sloppiness of these arrows. This is a lone phosphate, or an inorganic phosphate. It's inorganic because it's just phosphate. There's no carbon attached to it. But ADP, or adenosine diphosphate, has carbon in it, so it's organic phosphate. The electron here will combine or be used, really it's the hydrogens. We'll talk about that in just a second. The hydrogens are what are used to combine this inorganic phosphate to the ADP to make adenosine triphosphate, which will then be used in the Calvin cycle. But we're not going to talk about the Calvin cycle today. The Calvin cycle, or not in this video, will be discussed in another video. We're really talking about what's going on here and how these products come about. But what I want to do now is kind of focus in right here on the thylakoid membrane. Okay, so on this membrane, if we were to zoom in, we would see something like this. So here is the thylakoid membrane, and I know this is a lot that's here. I highly recommend that maybe you copy it down, pause it, copy it down as best as you can, and then add detail as you hear me continue to describe it. But what you see here, that thylakoid membrane is made out of a phospholipid bilayer. Remember these circles with the two lines? So it's a phospholipid bilayer. And what you might remember is that this inner part, right here is hydrophobic, right? And the heads of the phospholipids are hydrophilic. So this region right in the middle is all hydrophobic, which means that things with charges, things that are polar, can't easily get through those the phospholipid bilayer, which is really why these things exist. These are proteins that allow things to move in and out of the membrane that have charge or polarity or are simply too big to move. So I'm going to scroll up just a little bit here. All right, so we're going to start here and work our way across, and I'm going to explain a couple of things here. So when the light here is exciting the chlorophyll, these green dots are the chlorophyll that are really embedded within what we'll call Photosystem 2. In Photosystem 2, that's this whole protein here. It has these reaction centers. And so the light here is being absorbed by the chlorophyll, which is then exciting the electrons. And that excitation of the electrons is called photoactivation. That's a term you want to make sure you know, photoactivation. So that's how this light is exciting these electrons. Those electrons get bounced from chlorophyll molecule to chlorophyll molecule down into the center or the reactor here. And then it accumulates all this energy and jumps up to a higher state and is then passed off onto this protein that I've identified as PQ. Okay, PQ. So PQ stands for plastoquinone. And that, so plast is an important word here. Remember, this is a chloroplast. These are plastids. This is a type of organelle that has to do with sugar, and that's what that plast refers to. But this protein is hydrophobic, so it can stay within the fatty acid tails of the cell membrane. It can float around, and it sends these electrons. It continues to reduce future molecules as it sends it down something called the electron transport chain. And as these electrons are moving down, it creates actually a pull a pull on hydrogen ions or protons that are in the stroma, right? So the gel around the thylakoids, and will pull those hydrogen down through the membrane into the thylakoid space. Okay, so the hydrogens are building up here into high concentrations. But as these electrons are moving, they need to be replenished in Photosystem 2. And that's really where water comes into play. Water is the source of electrons for the Photosystem 2, and then for Photosystem 1 as well. So the water gets broken, it gets oxidized, it's losing its electrons through this photosystem. So when the light excites some of these electrons, it needs to be replenished, and it pulls the electrons from the water. And that's photolysis. That's another word you need to know. You want to make sure you know photolysis is a major part of light-dependent reactions. But the byproducts of that process are that it creates half of an oxygen molecule, because there's only one oxygen in water, and 2 hydrogen ions, or 2 protons, which happen to also, again, be in the thylakoid space. So we end up with an even higher concentration of hydrogen. All right, so we'll come back to that in... No, let's go ahead and do that part. So these hydrogens build up. There's a high concentration inside as opposed to outside of the thylakoid. And these ions, these protons, are going to try to find equilibrium. And to do so, they cannot go through the phospholipid bilayer. They have a positive charge, right? This is a nonpolar area. Anything with a charge is not going to like it, or it's going to get repelled, in fact. But these hydrogens find their way through this other protein called Atp Synthase. It's an enzyme that synthesizes ATP by taking adenosine diphosphate and inorganic phosphate and bonding them together to make adenosine triphosphate, which is one of the things that's going to be used later in the Calvin cycle. There's the Calvin cycle. All right, but we haven't finished the story. This is I'm going to write this one out. This is osmosis. Excuse me, let me make sure. It's chemio-osmosis. Chemio-osmosis. Spell that again. All right. And that's going to be something that comes back when we talk about cellular respiration in similar detail. So we've got these electrons that are excited now that are sitting in this protein that's hydrophilic, the PC, or plastocyanin. It's written up here, plastocyanin. And it's in this hydrophilic protein, which is not in the membrane anymore. It's outside the membrane. And light, again, is coming in and exciting what we will call Photosystem One. Okay? So Photosystem 1 is losing its electrons. It's being oxidized. It's losing electrons. But it needs to be then reduced by taking electrons from plastocyanin, which got it from plastoquinone, which got it from water. Right? So these electrons are just following along. Because there's a need to fill, they move forward. This light's exciting those electrons again, so we have some more photoactivation. And then this FD stands for Ferredoxin. Ferredoxin, which has iron in it, you might have figured that out. Ferredoxin is then going to reduce NADP+ and give it those electrons through this other protein called NAD+ reductase. It's another enzyme. We'll learn more about enzymes, but if you need to know more about enzymes, please update your Need to Know Next Steps document. But now we have reduced, because it's got extra electrons now, NADPH, which is another reactant to the Calvin cycle. So there's a lot going on there, and what I wrote up here is a little, if you got lost on reductions and oxidations, here's my little trick for it. Oxidation is loss of electrons, and reduction is gain of electrons. And really just trying to follow where the electrons go, and that's actually a lot of organic chemistry, is following electrons. I hope that makes sense to you. I don't know how long my video is. It's about 15.5 minutes. I'm going to stop here, and you should see some questions up here now, if you're an Edpuzzle.